Good morning, everybody, and welcome to our second workshop of the Living Soil series. This workshop is being presented by the Cape Town Together Food Growers Initiative in collaboration with Nemlabs and PHA Campaign. The Food Growers Initiative is a collective of anybody who wants to grow food, with a vision to create a food forest out of Cape Town to enable food freedom. It originated during the pandemic to connect the entire Cape Town and surrounds. Today, being our second workshop, we're covering plant parasitic damaging nematodes. And we're also going to be covering the soil health results of the 5 gardens that had the soil tests done. Today, our presenter is Sheila Storey from Nemlabs. Sheila holds a BSc Agri, majoring in entomology and plant pathology at Stellenbosch University. This was followed as a stint as a researcher at the Department of Agriculture, working on maize in Potchefstroom. She then transferred to Stellenbosch and worked on small grains, example wheat. After completing her MSc in nematology, she lectured at UWC in a course geared at crop protection. In 1987, she started the Nematode Diagnostic Laboratory, which now resides in Klutmutz. This is expanded to include labs in Upington and KZN, and she consults all over Southern Africa, covering all crops. In 2020, she took over the Soil Health Support Center, which now forms an integral part of her laboratory. And they also do insect IDs and have started a plant disease laboratory. Sheila travels all over Southern Africa doing presentations on nematodes and soil health. And we're very lucky to have her time today to speak to us. Thank you very much, Sheila, and over to you.
Thank you, Erica. Excuse the noise, it's pouring with rain here, but let's see how we can go. All right, so this is the second presentation I'm doing. The first one concentrated on soil health, but with my presentations I've done for Nazeer in the past in Philippi, I found that the one about nematodes on vegetables was actually what the participants were interested in. So we've decided to continue with that, and this, as I say, this is the second one in our soil biology workshop. Sorry, I've got it. Okay, so the contents for today, I'm going to tell you what a nematode is. I'm going to tell you what symptoms you're going to see on the vegetable crops, either on the roots or on the tubers. And then we're going to look at control strategies for these harmful nematodes. And then the last section is going to be the feedback for the soil health tests I did for 5 of the gardens, which Erica and Nazeer sent in. Excuse me. So to start off with, they're microscopically small. You cannot see them with the naked eye. They look like little snakes, although we call them worms. It's a euphemism, it's just completely the wrong term. So I suppose nematode is a better term. They vary in length from 0.1 to 1 millimeter in length. They vary in shape. You can see they can be very long little snakes, or they can be fat and round. The females swell up. They basically occur in your groundwater in the soil. You also get them in marine pools, but obviously we're just concerned with if I can put it that way, agricultural soil, so the groundwater in agricultural soils. And they feed on various organisms in the soil. But for this session, I'm going to concentrate on those that feed on roots, plant roots. So just to put it into a bit of a perspective in terms of size, look at the top right-hand corner. The horizontal line is the root lesion nematode. And some of you would have seen that in your results. The vertical line is a human hair. So it just shows you how small they are. If you go across to the left-hand side, In the case of the root knot nematode, that worm-like structure has swollen up. The female swells up, only the female. And if you can pick her out of the tuber, the potato tuber or the roots, and put it on your thumb, that's the size. And I've done it. It's quite an art, but you can do it. So they really, really are small. And that's why you have to have your analysis done at a laboratory. The bottom right-hand corner is a photograph of a sheath nematode on Swiss chard, taken in Franschhoek about 20 years ago by Professor Milan at Stellenbosch University. And there you can see the sheath nematode feeding on the root tip of the Swiss chard, just behind the growth point, and that's where it creates its damage. So it's an external, in this case, it's outside the root. The other two on the slide are inside the root. This one feeds on the outside, which makes it all a little bit more complicated. So feeding mechanism is, how do they feed? You would have seen there's a stylet. You can see it moving up and down. That stylet, it's a long, thin, hollow tube, which works like an injection needle. And then it injects a poison into the plant cell and the root, or it removes plant sap. So it feeds on or in the roots, and it's a chemical reaction. It's not a eating. Many people say to me, I can see the damage. You can't see the feeding damage. You can see the change that happens in the plant cells, and that I'll show you when we get to the symptoms. So there are different types, as I said. We're going to concentrate on the middle one, the herbivores. I spoke about the other ones in the previous workshop what we had, but we're going to concentrate on the herbivores this morning. Those are the ones you're more than likely to find in an analysis is the root lesion nematode. That was that thin one compared to the human hair. The root knot was the swollen one on the thumb tip. Then you get a stunt nematode. The sheath nematode was the one that you saw in the bottom right-hand corner. And then you get a stubby and you get a cis nematode. These are the ones you're likely to find, and which I did find in the analyses I did in the various gardens. Okay, so we look at the root lesion. That's the one that I compared to the human hair. In this case, the photographs are taken on potatoes. You can see on the left-hand side, I'd say the old people, it's the wrong term to use. But in the past, a lot of farmers would talk about the skin nematode. In Afrikaans, they talk about a skull album. It creates a strange, like a slight indentation, but a rough thing. But please don't confuse it with the lenticels, which you see on the right-hand side. The picture above that is a picture I did in my master's in 1985. That's the root lesion nematodes inside a maize root plant. So what happens? It blocks the uptake of water and nutrients, and nothing can move through that root. The root knot nematode, this is on cucumber. So let's start in the middle. That's the picture I showed you. So on the left-hand side is a cucumber plant, the roots, which have been infested by that root knot nematode, and those are the symptoms that are caused. So we term them giant cells. Remember, I said it was a chemical reaction which takes place inside the root, and it results in those galls. On the right-hand side, you can see they're very small, little round bubbles, as it were, where the galls are starting to form. There's also one in the middle of the picture, which looks like a little point. But in the extreme cases, you can see something as it is on the left-hand side. If I look at beetroot, this comes from the Philippi area where we flattened, or the producer flattened a sand dune where there was a lot of Port Jackson on it. And I've got to mention it, Port Jackson is a host for root knot nematode. So if you're going to remove Port Jacksons, you can expect this nematode probably in the second or the third year. And this is what the beetroot looks like. You get those little galls, and then it can form, as you can see in the top right-hand corner, that very rough, almost callus structure on the beetroot. But that's in extreme cases. But these roots are compromised. They cannot take up water, and they cannot take up any nutrients either. This is what carrots look like, the root knot nematode. It gives you a misformed carrot. And then you can just see on the tips of those roots, the thickenings or the little gauze. It often looks like beads, which you've threaded on a string. Yeah, so that's root knot nematode on carrots, which makes it difficult to market, or you can even get this size, and it doesn't grow much larger than that. But this is again in extreme cases. On potatoes, I showed you the Lesion nematode. This is what the root knot nematode does. It forms like these wart-like structures on the potatoes. And that top right-hand picture is the female right inside the tuber. And she can sit there as well. She's usually on the outside, but that photograph, it was just, I cut the potato in half and I stained it with beetroot salad juice, as it were. And then the potato goes pink, and then the nematode is like almost a clear white. So at the bottom right-hand corner, you can see the galls on the potato tubers, and those are extremely difficult to market. Nobody wants to buy potatoes like that. They're quite edible. They're a little bit sweeter, and they've just got a lot of extra protein in them. This is root knot on tomatoes, and you can see it doesn't really form galls in this case. And I don't know why. It just forms these very thick roots, yeah, which is quite a problem on tomatoes. The tomato will often grow, and then the tomato, you'll see it, it'll be perfectly nice. You can see the bottom left-hand picture, they're green, but they don't ripen. That root system is so compromised that the plant then starts dying, and you don't get the tomatoes to go red and to ripen. This is the Stunt nematode on Brassicas. The bottom picture, the bottom right-hand picture is what I saw on the farm. This is a number of years ago in the Suntveld, which is the area very close to the Lambert's Bay on the west coast. And we went out there, and what happened, you can see in the bottom left-hand picture, it didn't form a head. In the case where there were cabbages, it didn't form a head. You just get these loose leaves, which is quite frustrating. Yeah, that was quite devastating, that contamination we had there. I've shown you the sheath nematode on spinach. You also get it on carrots. It's quite common in the Philippi area and in the Joostenburg Vlakte on carrots, the sheath nematode. It causes the carrots to split. They also talk about it's got teeth. It's a typical symptom as well. The stubby root nematode is very common on the crops I've listed: maize, carrots, onions, cucurbits. Those are your melons, your things like what else? Your pumpkins, and obviously your cucumbers as well. You can see the photograph of the sweet potato. That was a discovery I made in 2002. That was also in the Suntveld. We thought it was insect damage, like a beetle or yeah, but it wasn't. In the end, it was nematodes, this particular nematode. It prefers a very coarse sandy soil. You can see in the bottom right-hand picture, it's got this curved tooth. It almost looks like a cigar. It's quite a big nematode, so it battles to get through finer soils. And then, as I said, it's quite a problem on carrots. It just cuts the carrots in half. They just stop growing. And then eventually they will start getting these thin roots growing out of there again. This is the Cis Nematode. It's sort of an insignificant little nematode, and then it causes these brown dots, which you can see, and those are called cysts. They are throughout the Cape Flats. They were brought into the Philippi area many, many, many years ago. If you're really interested, you can go and Google sugar beet docking disorder. It's a common disease in Europe, or a common nematode, should I say. And it is a complicated nematode, and that you cannot control it. But I'll get to that point later on. And this is Nazeer's problem. He has quite a few of them. And it bursts through the roots, and then it causes damage. It works very differently to the other nematode. And it's coated, and this is why it's called a cis nematode, and that can remain in the soil for, in this case, for this particular nematode, for up to 8 to 12 years. It can remain in the soil until you plant a host again. So to look at the results that I got, I've listed the farms on the left-hand side and the nematodes across the other side. Root knot was common throughout, but not in very high numbers. That was the one that forms the galls. The stunt nematode was the one that I showed you on the cabbage. There was a little bit throughout, because I know you all grow a lot of cabbages. The stubby root was also found. The only farm that it was a problem in high enough numbers was in the Boerkaap garden. And that is why I've asked them to treat. If you go to the top, to Verkoop, Nazeer's farm, the Cis Nematode numbers were too high. We've got a problem there. We're going to have to address that. I sent Nazeer an information sheet on the Cis Nematode. We're going to have to work through that very carefully. Okay, so how are we going to control them? Sorry, let me just go back to this slide with the root knot nematode. It's going to build up. We've got to keep an eye on it, but I think you're doing a lot of the things you're doing correctly, and hopefully it won't. But that's the one I'm really concerned about. So what you have to do is you have to rotate your crops. Don't plant the same crop over and over and over, but I think you know that. And what I mean by that is it's not only a cabbage after cabbage, it's also brassica after brassica. So we've got to be very careful. And, you know, like don't grow a tomato and a potato and a brinjal one after the other. Try and put something else in between. You've got to use resistant cultivars. That's a little bit difficult because the only ones that I'm aware of are tomato cultivars that are resistant. So don't plant Moneymaker or Floridade, but go for different cultivars if you're growing tomatoes. Please use clean plant material. I can't stress this more. So I showed you the root knot and the root lesion nematode are inside the roots. So if you're going to bring plant material from outside or even your own, which you've made, and it's already got the nematodes inside the roots, you're just going to put it straight into your new garden and you're going to undo everything that you've done. Be very careful. Use preferably, I don't quite know what material, coir or something. Be very careful of buying compost unless you're sure the compost is clean. Avoid potatoes which had those tubers on. Literally go and burn them, throw them away. Don't put them into your compost heap and don't feed them to the animals. Those nematodes go right through. Unless you can reach a temperature of 50 degrees Celsius in your compost heap throughout the heap, you're going to have a problem with those nematodes. I showed you the tomato roots. So if you find something like that, the cucumber roots, remove every single root. Even if you have to sieve it, remove it and burn it. Because they're just going to remain there in the soil, inside the roots, and you're just going to plant your new crop in those, and with those nematodes present. The other thing I've also seen, which a lot of commercial farmers do, is they leave the potatoes in the soil because that's fine. They can store them there until the price is right. But remember, the nematodes increase all the time. And then in the end, you're going to have a bigger problem. So if you've looked at your roots throughout the season and you in any way see nematodes, whether it's on the potatoes or on the roots, but this only applies for the root knot nematode, which causes the galls, lift it as soon as you can and sell it. Don't keep it in the soil. Biofumigation, Ibrahim has spoke about this. I'm going to go through a couple of slides, how you can use brassica plants to clean your soils, and not only for nematodes, but I'll discuss that later. I'll also discuss in detail what we call solarization. Here in the Western Cape, we're lucky we have very hot temperatures, and we can create those increased temperatures in the soils, and we can kill off a lot of the soil-borne diseases, which include nematodes and Phytophthora, Pythium, Fusarium, all of those. I'm going to talk a little bit about biostimulants and manure. I'm going to mention biologicals. There isn't really anything on the market that works. And then I'm going to touch on chemical control. Okay, so we're going to rotate crops using different plant types. I mentioned that, and as you've heard, I've been talking about different nematodes on different crops. So the idea of the rotation is to limit the buildup of the plant parasitic nematodes. So use a good rotation system. Include brassicas as much as you can with the others, particularly if you're going to be planting maize and tomatoes and potatoes and brinjals and cucumbers and watermelon and spun speck. You've got to start adding brassicas into that rotation. If you're going to leave your soils fallow, in other words, with nothing on top of it, I don't like it. You've got to remove the weeds. Remember, the weeds are also hosts, particularly nightshade. You know, I don't know what weeds you have, your Port Jacksons. If they come up, remove them as soon as possible because they are hosts. I say there, apply chemical control on rotation crops, and that's not really an option for you in the small areas because the chemicals are very toxic and you're not going to be able to buy them. They're not going to be feasible. They're also very expensive. So the crops that if you've got root knot nematode, I've just listed them all here. Maize is an excellent host for root knot nematode and root lesion nematode, but maize does not show gauze on the roots, and I cannot explain to you why. It just doesn't, except in severe cases. So not too much maize, please. Potatoes, tobacco, peppers, obviously not tobacco for you, but peppers are also hosts. Your cucurbits, your melon, watermelon, butternuts, and the old pumpkin is not so, is not so badly affected by root knot nematode. For some other reason, the old wit pompoen is a better option. Sweet potatoes, peas, beans, carrots, they're also excellent hosts, and obviously there are many more. Look at the weeds that I said. I've mentioned Port Jackson and nightshade, and so I really don't know what you have in your area. Hopefully you don't have any weeds, so we've avoided that host. I've put this table in. I'm not really going to go through it. I've tried to list the crops that I saw. I think you must just keep this in the video presentation and have a look at it. But basically, a lot of the crops are good hosts for a lot of the nematodes. If you've got root knot, you can look at your brassicas and you can look at onion as a non-host. And your small grains, but I don't know if you're going to be planting things like wheat, which is, or oats, which is an excellent non-host for root knot nematode. I've put this in with weeds. I'm not going to go through it. If you've got a lot of dibblekey, then you don't have a problem with root knot, but the other information is non-existent. We just don't even know. Very few people work on weeds in South Africa, in terms of nematology anyway. You can, as a non-host, I mentioned a couple, but for a small garden, you can use marigolds. They're excellent. What they do in Kenya, they plant a row of marigolds, then they plant the vegetable, a row of marigolds, and a row of vegetables. Next year, you plant your vegetables where the marigolds were. And you can use exactly the same with the wild rocket or Arugula sativa. It's very popular if you're going to Woolworths in their lettuce mix, then wild rocket is included. You've just got to find a very good market. But restaurants, so if you're going to, you can be very entrepreneurial and include wild rocket. It's an excellent non-host, works very well. So that's a consideration if you're planting lettuce, you can include wild rocket. The clean plant material, I've mentioned this and I've shown you the photographs. The root knot and root lesion nematodes are what we call endoparasites. They're inside the root, so please make very sure that your plant material is clean. Seeds don't carry these nematodes, but obviously if you're going to grow seeds in infested soil, you're going to get it in the root. So try and use a medium to grow your seeds in which you know is clean. If you do have these infested roots, please, as I've said, remove them, burn them, do not add them to the compost, and do not feed them to the animals. They go right through the rumen and come out at the other end, and particularly if you're going to use the manure. So by leaving tubers in the soil, I've mentioned this, the farmers who leave potatoes to store them, it's a cheap storage, and also they don't have to, they wait until the price is right. But please, if you're going to plant potatoes, look at them And the minute you get these gores or warts on them, lift them and yeah, sell them as soon as you can, because the symptoms are only going to increase. So to look at biofumigation, this is an option. This is a lot of science. I'm not really going to talk too much about it, but your brassica plants have a component called glucosinolates and an enzyme myrosinase. If you chop them very finely, the roots, and actually more it's not really the roots, it's the above ground parts, your leaves and stems. If you chop it very finely, in other words, you break the cells, it releases these isothiocyanates, and then you add a bit of water, you incorporate it in the soil, and it will then kill most soil-borne diseases. There are one or two exceptions, but it's quite an intricate process, and so I'm going to go through it. You do your soil preparation. Your planting time is, you've worked out when it is, your sowing density, your sowing depth, you've got everything right, and you've added a bit of fertilizer because you want quite a standoff this biofumigant crop. But I'm going to ask you to use cabbage. That's probably the best one, or any for the brassicas for that matter. The more above ground biomass you have, that's leaves and stems, the more cells you have. The time of chopping is also quite important. You can harvest, but preferably harvest a little bit early. You've got to break as many cells as you can, so it's very finely chopped. And then you've got to incorporate it into the soil very quickly. So don't do too large an area at a time. You can add a little bit of water once you've incorporated it, and then that gas will be released. These are the crops that you can consider. This is what I use for producers on a large scale, but in your case, cabbage or cauliflower or Brussels sprouts, any of those will work. So this work that I'm going to show you now was done in Botswana by a master's student, and she planted cabbages, she harvested what she could, but you've got to leave at least 4 to 6 kilograms per square meter of material, which is quite a bit, I must admit. You chop it as finely as possible. Labor, you've got to get the children to chop or do something. You work it into the soil as soon as possible. You add water, and then you leave it for 6 weeks. And that's critical because if you plant sooner than that, your plants are going to die as well. It's quite a strong chemical which is released by the cabbages. But remember, those cabbages, those leaves will also provide you with green manure. And then you can plant your summer crop. So it works very well. This is what we tried it between vines. We obviously didn't incorporate it because it would have killed the vines. But this was done just to show you what it looks like. You flail it and thrash it, and then you start chopping it as finely as possible, and then you work it into the soil. Solarization is the other option. We use sun energy. It's only plausible for the summer months, obviously. We do this a lot up along the Orange River where there are large tomato producers. You use a see-through plastic. It's almost white. It's not quite see-through. It's a 30 micron plastic because it's got to last for 2 months. You cover your soil with it, leave it there for 2 months, and you try and raise the temperature under the plastic up to 50 degrees Celsius. It'll kill weed seeds, fungal spores, bacteria, nematodes, and anything else that's in that top layer. Obviously, only up to where the 50 degrees Celsius gets to. And this is when you start getting damping off or nematode problems, then solarization is an option. The chemical control, I'm not going to suggest that you use it. As I said, it's highly toxic and very expensive, and I would not use it in the areas where you're planting. It's just not, it's environmentally and just on the human scale and animal scale, it's not viable. You can't do it. You can use Jay's fluid. I know they're not very fond of me giving this recipe, but I do use it for any gardeners. Prepare your seed beds as if you're planting. Measure a 1 square meter area. Mix 50 milliliters of Jay's fluid in 5 liters of water. You can just do it in an ordinary watering can. Then you spray through the nozzle and you drench that 1 square meter with a full 5 liters. of water or mixture that you've got. It is a drench. You leave it for 2 weeks, rake it over quite thoroughly to release any carbonic acid which is left, and then your soil is ready to use after that. Also quite a successful operation you can do. I'm going to mention this in terms of what we call root or biostimulants. We also refer to them as PGPRs, or plant growth promoting rhizobacteria, and this you will find in the market as a bacillus or a mycorrhiza, trichoderma, kelpac, things like that. All you're going to get in this case is improved root growth. But it is very helpful if you've got a very low population, which a lot of you had, of nematodes. And it's a numbers game. So if you've got numbers, these additions will suppress the nematodes. But obviously, if you've got very, very high numbers, this is not going to help anymore. But you can also do things like carbon supplements, which you're doing in the case of manure and mulches, compost tea. That will also help. I think most of you are doing that in any case. So final point on the biostimulants: stronger root systems, the plants have less stress, stronger plants in marginal or poor soils, and you're going to have fewer nematode problems, very definitely. And you've put all those other control systems like rotation, etc. in place. Erica, I finished on the nematodes. I don't know if you want me to go back to start the soil health feedback, or if there are any questions on nematodes.
I think there's a couple of questions on nematodes. Hafiz Sonday, you've got a question? Would you like to ask your question?
Hi, good morning, guys. Hope you guys are all well. Sorry, I joined a bit late because I was struggling to get through to the link. But when I joined, there was some relation between root knot nematode and Port Jackson. Can you please tell me what that relation is again?
Yes, Happy. So what I was saying is that, did you see the beetroot picture that I had?
Yes, ma'am, with the nodules on it.
Yeah. That's correct. So all plants are host to nematodes. Just because we think it's a weed doesn't mean to say it's not affected by the nematodes. So Port Jacksons are a good host for root knot nematode. That picture that I had of the beetroot was a farmer who flattened a sand dune in Philippi and then planted beetroot. And 4 months later, he had that problem. I've actually had Port Jackson roots in the laboratory to confirm it. So what I'm saying is, if you've got a Port Jackson tree, or it often makes like a ground cover, and you remove it, you will have an infestation in that soil. You might be able to get away in the first year, but by the second or third year, you're going to start picking up problems.
So then do you suggest that we leave the Port Jacksons in the ground?
Yes, you can. Or you remove it completely, roots and all. And then you'll have less. Look, that guy just plowed it over. He just flattened it.
Oh, I see.
He didn't actually physically, but I mean, he's not going to do that on a large scale. It's just easier to flatten it.
Yes. So if you do remove them and perhaps you don't get all the root systems out, you could then maybe plant some white mustard along that area and then chop them down and biofumigate. Will that help?
Yes, you can do that.
Okay, cool. Because we have Port Jacksons on our periphery that we are reluctant to get rid of because they provide a nice cover for what we're doing on the farm. So the thieves can't see all the things we're growing because they like to come shopping in the night.
No, that's fine, Hafiz. Keep them there. I don't have a problem. Just be careful of the roots that'll spread from that Port Jackson into your garden. You know, you could put a barrier there or whatever, but yeah, just be careful of that. Okay.
Cool. So if you do end up removing any Port Jacksons, it's best to remove the complete plant roots and everything.
Absolutely. Yeah, it's a little bit of physical effort, but it's got to be done.
Yeah. Cool.
Okay.
Thanks, Hafiz. There is another question from Sheila. Please advise what soil mix would be good to redo my containers to avoid nematodes from the start.
Well, Sheila, I'm not quite sure what you mean by soil mix. Are you talking about a purchased mix, or are you talking what you have? You know, are you removing soil from an area and just putting it into your containers?
Hi, good morning. I mean, even a soil mix that I can buy, not from that I'm taking it out from somewhere. I grow for home consumption in baths and containers, and I do sometimes want to redo the bath. And what I do is I don't always remove all the soil, but if I want to try and make sure that I start the bath from fresh, what would be the best?
Well, Sheila, on the market, there's very little that's good. I'll be very honest with you. We've done some tests. It's what you purchase in the nurseries is really not worth it. What I would do in your case is spread that soil out on an area of either a piece of plastic or a cement block.
Yeah.
And dry it out and heat it at the 50 degrees Celsius, or try and cover it with plastic, or that's what I would do.
Then I can reuse it.
Or you can plant the wild rocket. It's quite expensive, or marigolds. Plant quite, you know, before you remove the soil from the container, is plant quite a lot of marigolds that you can get the roots throughout that soil and kill off whatever's in there. That's also going to help you.
Thank you. Thank you.
Thanks, Sheila.
Sorry?
There is one more question from Jill McKay asking, does solarization and Jay's food also kill the beneficial microorganisms? If so, what do you recommend to restore biodiversity to the soil?
Yes, the solarization and the biofumigation will definitely do it. So that's in extreme cases. The analogy that I can use here, you know, you've got cancer and now you're trying to do vitamins. So sometimes you don't have an option. But those are short-term, quick fixes. But by planting marigolds and wild rocket, you know, then you're not going to be affecting the biodiversity. It's kind of going to be, the answer is in what your position you're in. That's in the end what's going to determine what you do. Is that what you wanted to hear, Jill? I'm not quite sure.
Yeah, hello, Sheila. Yes, that makes complete sense. It's a bit like taking an antibiotic, isn't it? Where you kill the bad and the good, and then you, I suppose, but sometimes you have to do it because it's a life and death situation. Would that analogy work? Yeah. Thank you.
No, that's exactly right, Jill. It is, unfortunately, you get to a point you didn't recognize it for whatever reason. You know, these things happen. You could have brought it in from outside with bad plant material. At the end of the season, you see, I can't plant the next crop, and you have to. You don't have time to put a marigold in. So then afterwards, to restore your biodiversity, the best way to do it is with a diverse cover crop. Again, that's going to depend on the situation you're in. Or then you put in manure and compost and that sort of thing. Just feed it quite well.
Great. Thanks a lot, Sheila.
Okay.
Thanks, Sheila. A question from myself. What cover crops would be good to plant when you're trying to control root knot nematodes?
Okay, I mentioned that in a slide, Erica, the one about the marigolds, the wild rocket, sun hemp, or even any of the oats, what we call sire oats, black oats, or wheat, brassicas, all of those will control a root knot nematode. By cover crops, it's usually, if I think of the farmers on a large scale, they would put a mix in of a radish, a small grain like wheat, or I'm trying to think what else they put in. We try and put a legume in to get nitrogen fixation. So you've got 4 or 5 different plants. But again, you can only do that, and it's a long-term solution. You know, if you have to plant something this season, then those kind of cover crops, it's not an immediate control of the root knot nematode. But in small plots like you do, the marigolds work quite well, or wild rocket. One should also, if I look at the Kenyans, they've got into a habit of a row of vegetables, a row of wild rockets. The only problem with, you know, you're losing quite a lot of space because you can only really produce on half your property, unless you can find a way of selling your wild rocket. You've got a market for it. You know, you've got to try and find one. And then it
Sheila, I was thinking more along the lines of where you're going to leave the ground for a season and not grow a crop that you're going to harvest. So would things like clover work well there? Because you said you want to do nitrogen fixing.
No, not clover. Rather go for an oats. Yeah.
Oats,
Oats
okay.
will probably grow best. The black oats will probably grow best in your the, I don't know. I mean, I really don't know about cover crops. But sun hemp, I know, is a thing that will only grow in Natal and Zambia. I know, but you know, I don't know if it's going to grow so well in the Cape. I would just use an oats. I know it's going to work in a Mediterranean climate, or wheat for that matter.
Great, thank you very much.
What I can do, Erica, is you people can come up with an option. You know, whoever you've tried something, just don't plant maize, whatever you do. If you come up with something else, phone me and I can say yay or nay.
Okay, great. Thanks very much, Sheila. And Hafiz has got another question.
Yes.
One quick question. So the arugula rocket, the marigolds, and the white mustard, they can be good deterrents for nematodes. Are they able to also provide that function without chopping them down and intercropping them with your already growing crops? Is it possible?
You've mixed the crops, Hafiz, so be careful. So the marigolds, the wild rocket, are crops that what they do is that they either take the nematode in and then it doesn't multiply. In the case of the white mustard and the cabbages, you have to break it up. It doesn't work any other way.
I see.
So the marigold and the wild rocket, well, the wild rocket to a degree don't have that methyl isothiocyanate, the ITC.
Okay, so basically you can intercrop the wild rocket and the marigolds as like a trap for yes, nematodes to deter them from your actual growing crop roots.
You can, but it's difficult to, you can intercrop it, but the Kenyan way where you do a row is a little bit, it's easier to manage.
Yes, but on a small, when you're on a small scale, it becomes a bit problematic because you limited in the area that you have, and so your production is also limited, and therefore your income is also limited. So I'm just trying to find a way where you can do all these things together at the same time. You know.
No, absolutely, Hafiz, but you're going to have to try and see what works for you.
Yes, for sure. Trial and error, for sure.
Absolutely, yeah. Okay.
Sheila, we have a question from Abida, which was one of the gardens that you did the test on from the Burkhardt. She says that she's using marigolds and wild rocket, but should they be planting more to improve the soil web structure index?
Sorry, just repeat, Erica. It's quite a complicated question and answer that I'm going to think about. Sorry, what did she say exactly?
We have many marigolds and wild rocket. Should we plant more to improve our soil web structure index?
Yeah, okay. I can't remember what it looked like. The soil, that's in the feedback. My problem was a beta in the boer carp had a nematode problem, and that was a stubby root nematode. And that's a different nematode to root knot, very different. And I don't want you to put too much wild rockets and marigolds in. If you're already using it, keep it. I'm going to have to address that later differently, but just let me get to soil health. I can't remember what was in that. I'll get to that point.
Okay, no problem.
Sorry,
thank you.
Yeah. Okay.
And then we had a question from Sophia. Sophia, would you like to ask a question?
I think we've also got sheath nematodes on our spinach, and it wasn't reported in that lab thing that you sent, because we do have that rusty spots on the spinach as well.
Okay, Sophia, if you have rusty spots, that is more than likely a bacteria or a fungus. I don't know. I'm not that familiar. We do have a laboratory, a disease laboratory running now as well. Yeah, well, you can either, I don't like to do it, but you can try and send us a photograph on WhatsApp. I don't like doing WhatsApp diagnosis because, yeah, you often miss out on something if you do that. What happened, I'll have to go and look at the sheath nematode. I've got a poster in the laboratory with the sheath nematode on the Swiss chard. You get almost thickened roots as well. They don't form galls, but you get thickened root tips with the sheath nematode on Swiss chard.
Yes.
And
Thank
the rest
you, Sheila.
is an above ground, either fungus or bacteria. I mean, I did plant diseases 40 years ago, but I can't remember what I studied. But we have a plant pathologist in the laboratory.
Thank you.
Okay.
And one last question from Sheila again, asking, will rows of alfalfa between the veg work the same as marigold and wild rocket?
No. Sheila, in some cases, this gets a little bit technical. In some cases, the alfalfa is a host for root knot nematode. There isn't only one kind of root knot nematode, there are quite a few. And there are differences in the alfalfa cultivars. I can't remember. Sheila, what was your farm? Was it in one of the tests?
No, Sheila, mine wasn't in one of the tests. I'm again the one that grows in containers.
In pots. Sorry, you're in pots, in containers.
I was just wondering because I actually sprout my alfalfa, and you know the ones that doesn't sprout, I always just put it in the ground and it starts to grow, and yeah, and then there's an alfalfa plant and then it will seed. So I was just wondering, what does that do if I have that in my between my veggies? Will it bring more nematodes, or will it be any beneficial in there?
Yeah. Well, Sheila, you've caught me. I don't know. Because
Okay.
the alfalfa that you're talking about is not grown on a commercial scale. You know, out there. Talking about farmers who grow alfalfa for cattle and sheep. And then it doesn't grow, and it is often nematodes, which causes the problem. I don't know in your case. I know what you're talking about.
Yeah.
I'll have to get back to you.
Thank you.
I will find an email address from Erica, and I'll see what I can find. I can't promise, but no, I don't know, if nematodes and which nematodes affect the alfalfa that you're talking about. Sorry.
Thank you. Okay. I'll forward my email to Erica. Thank you.
Okay. Thanks for Sheila.
Thank you. And Sheila, that's it for now. You may continue. Thank you.
Okay, so what we did before I go into the feedback is I offered 5 farmers. We did the nematodes, the damaging ones, which you've just seen. But because we did a whole soil health talk, the previous talk was all about soil health and soil biology and reverting it to soil health, I offered 5 samples, which the laboratory did for the CTT Food Growers Initiative. And these are the results which I promised I would work through. So let's hope it makes sense from here onwards. Okay, so at the Soil Health Support Center here at the laboratory, and you can look at that website, we offer various tests. The first 2 are also done in the laboratory. It's no longer called the Solvita, we just call it the 24 CO2 burst test, carbon burst test, and we do the aggregate stability. The following 3, the Haney, the Slan, and the Millich, are done in America. They're quite pricey. The Mycorrhizae, which is a fungus which grows inside your roots, and I think I spoke about it last time, and then expands your root area, as it were. It grows outside between the cells and inside the cells. We do that too. We do a mycorrhizae colonization test. And then we do the nematode soil health analyses. And that's what I offered the 5 farms. And that's what I'm going to give you the feedback on. So those of you who have them in front of you, let's see what we can get out of that. So the first result that we get is a biodiversity, where I tell you which feeding groups, I tell you how many of each, and I tell you what percentage, and then I'm going to work through that for everybody now. Okay, so this is quite a complicated table. I'm going to start at the bottom with the totals. And the first reaction I had is those totals are extreme, except for Nazeer's. Even that, we never see numbers like this, not in commercial agriculture. And there are lots of reasons for that. My first reaction was, you've got too many. And I'll work through that now, and we'll see as we go along. Okay, so we look at the plant parasites. The counts in the plant parasites are going to be different to the analysis I did, because we use a different extraction technique. Nazeer and Abida and Fawaz too high. We've got a plant parasitic, a damaging nematode problem, which is what I showed you in the previous session. Abida, the problem that I have there is a nematode that came out, which you don't usually find on vegetables. It's usually on vines and peaches. So that's a bit problematic for me. Don't know where that came from. The next one I want you to look at is the bacterial. And you find the bacterial feeders in a nitrogen-enriched environment. The plants and ocean view, and even to an extent, Mangalisa, you've got too many. And I think that you're putting in too much manure, or it's not composted enough. Whatever you're putting in, it does get broken. Gets broken down by the microbes, but I think we must just go a little bit slower. But I don't know when you put it in compared to when you took the samples. So if you took the samples immediately after applying whatever you did, then perhaps you're going to get high numbers like that. But I've got a feeling we're going to have to change that a little bit. If I look at the fungal, not enough. There's not enough dry material going into the mixes. And be that, straw, that sort of mulch, I think we're going to have to start changing the relationship between the bacterial feeders and the fungal feeders should be more or less the same, and it's not. So all of you have got too little dry brown material, if I could put it that way, that's going into your compost or your mixes or your manures. I'd like to see a bit more. Having said that, in the omnivores, or the omnivores who feed on organic material, that looks good, which is a little bit at odds to the fungal, but we're going to have to change the fungal numbers. The predators, so I'll have to work out, I didn't put the slide in here. Predators are few and far between. I'm not so surprised that I didn't see them. I was surprised to see them in the case of Vachkop and Boerkaap. The only assumption that I can make, and that's an assumption you would have to let me know, is that those two have been farming for longer and their processes are a little bit better. It's good to see, Nazeer, that you've got quite a lot. That's quite high. What worried me is the root exudate feeders. It's a group that feeds mostly on root exudates, which are an indicator of stress in the plants, which we go back to the bacterial feeders. There's probably too much nitrogen, or the nitrogen-enriched source is too strong. I don't know if that's the right word. I've seen this when I've seen people put raw chicken manure into the soils. I've seen high numbers like this. But, and I've now said all of this, I seldom work with this kind of garden setup. You know, I usually work on a large scale. But that's the feeling. It's kind of around thinking, you know, we've got, maybe you must change, but it's difficult for me to say. I would like to do a second set perhaps in a year's time or at a different season. But as I don't know what happened exactly prior to taking these samples. So I'm saying all these things, I'm going to have to get feedback, but that's my reaction when I just look at these numbers that are in front of me. Okay, so what I did here is I took those numbers I've just shown you and I put it into percentages. And if I look at, we can start with, sorry, I just want to see what I've done. Yeah. So what I'm going to do, if you look at, for example, at, we can start with Nazeer's one at Vachkop. Your plant parasite percentage is too high, your bacteria is too high, and your fungal is missing, and the root exudate is too high. We're going to have to change that. A bit of the same for you. Too many plant parasites, too many bacterials, and too many root exudates. It's kind of forming a pattern in those farms. For was your bacterial count was hopelessly too high. It made out 70%, 78, almost 80% of your population. You've got an imbalance, and we're going to have to correct that. But the rest looks good. It really looks good. I would, in your case, just change the bacterial to the fungal. So you're actually an example of what it should look like, but by adding dry material into your compost or that sort of thing, I think we're going to be able to change that. Yeah, so yours looks good. Sophia at Ocean View, exactly the same. Too many bacterial feeders, and we could push up the fungal feeders, definitely. Mangalisu, the same, also very high bacterial feeders, and your root exudate feeders are too high, very definitely. I will put in dry material. So if we look at the ecological indices, and I'm not even going to go through that slide, I'm just going to go straight to what I found. This slide goes with the next slide, actually, but I'm just going to go through the numbers. So if I look at Nazeer, your structure looks really good. It really does. Your enrichment means you've got too much food for the microbes. It's still got to decompose quite a bit. It's got to go down to 50%. The structure in the basal index must be at 100%, then it's best, in green, but the enrichment is at 50. Better, we'd have to push up your structure index, very definitely. We need a few more alternative crops, you know, your rotation or your mix, we can increase that. Your enrichment is a little bit high, it's got to break down. For was, yours looks good except for the structure index, but I'm assuming there's still a way to go in that farm. Ocean View, enrichment is very high, but your structure looks good. Yeah, and then Mangalisu's looks the best of all. Yeah, actually quite good. I would just improve your enrichment, so by adding a little bit more manure or compost. It needs a bit, it's probably worked through, and now it adds an addition at this stage. The problem with these indices, remember again, I say, I don't know what manure, or did you add something to the soil recently? recently. So you've got to see it in the light of that. If you added something, a nitrogen enriched material, be it the compost or be it the manure recently, then your enrichment index is going to be in the red, very definitely. I've gone through them all. So this was the profile result. This is what it, you know, just an example, but, oh, sorry, I didn't want to put that slide in. Okay, so let's start. Verkoop is right up at the top in sector B, and that's excellent. Well done, Nazeer, you've had an opportunity to do it. Workcarp, you need to move to the right. So we're going to have to start adding a little bit more, a greater diversity of plants and perhaps a lot of dry material to that one. Class is the same. Ocean View are doing quite well. They can also move to the left. And at Mangaliso, at Ikamvaleto, we can move up and across to the right. Okay, and as I say, how do you do it? So I think you've all got enough enrichment, your nitrogen enrichment, which is the manure or compost, which isn't, you know, it's still got too much activity in it, but that can happen in the soil once you've added it. It can happen in the soil, so don't look too carefully. Except for Ecovaleto, you're all above the 50%, so you're in the right enrichment side, but you all need to shift to the right, except for Nazeer. He has a secret, which I don't know what to do, except that he's got Cis Nematode. And I don't know how we're going to solve that one.
Lots of compost, compost.
Yes, Hafiz, but the problem is it needs, with Nazeer it's worked, or if you've been doing it, you probably have enough dry material in there. The others, I would like to see more dry material included in the compost.
I see. So what we've been doing is telling the guys at the local dump here to send all the people with their garden material to the farm, and then we break that down and put that into our compost piles.
Yeah, you can put stuff like, if you're growing maize, I'm asking you not to plant maize, but those dry leaves. What the big compost is, a huge compost facility in Ceres, just outside Ceres, and they were bringing stuff in from outside, and it was the carbon footprint and the expense was just too high. So what they started doing, there's a lot of onion growers up in the Kobokkeveld, and they started bringing these onion leaves and put that into the compost to get it more fungal. And that worked very well.
Okay.
So think out of the box, think dry material. Yes, mulch, that's what you've got to start adding. And then you're going to shift it across to the right-hand side. Okay, and with that, I'm finished. Erica, it was quite a lot to work through. Thank you for the opportunity. To Nazeer for the opportunity. He always, presses on my number. Thanks, Nazeer. I'm also learning. Don't worry. Yeah, so that's what to show you the pictures on the left-hand side of the screen. That is the mycorrhizae. In the very top is, we call them arbuscules. They're like little tree structures inside the cells. The picture below that is the vesicles. They have storage organs, which are on the hyphae, which is also between the cells. And in the bottom picture, you can also see it's just you've zoomed out of it. Yeah.
Thanks very much.
Mangaliso here.
Yes.
Mangaliso, please go ahead and ask your question.
Thank you very much. I wanted to say thanks for the advisement and the test provided. I shall abide the instruction, and I will see what we'll make out of the advisement. Thank you very much for your help.
Good luck. I hope it
Thank
goes
you.
well. I admire all of you. It's not something I would do, but well done.
Thank you very much.
Sheila, thank you. Sophia has a question.
I just wanted to also respond by thanking Nemlab for the opportunity. And I think that I have so many questions. I think it would be best for me to send an email, Sheila, because there are so many things that I need to know, and also to ask, for changes. So thank you once again for the opportunity, CTT GFI, Food Growers, for the opportunity of our garden being selected. And thanks, Sheila.
Pleasure, Abida. You can send me mails. It gets a little bit busy in the lab, so if I don't answer you straight away, don't be concerned. Yeah, it's quiet in the winter, but the season's going to start any minute now, and then it's thousands of samples we have to get through. But I'll try my best. I don't always have the answers because I work on large production scale, but we can search and find something. Okay.
Yeah, we will definitely start on all the advice that Sheila has given us. I'm going to start as soon as possible. Thank you so much.
Okay, fine. Thanks, Abida.
Sophia, please go ahead.
Oh, yeah. From today onwards, the farmers on this farm has quite a lot of homework to do and work to do. So we do feed our heavy feeders with fecal pellets and with the Brassicas. When we terminate, we put bone meal in the soil. And when we do our compost, we add the ground manure to the compost, but we also put it like in between the arms, in the pathways. So when we do the no digging, we just put the ground manure onto the bed and we fix the bed for the next planting. So is that the right procedure? Because we don't really know how much to feed or when to feed. If it's a heavy feeder or nightshade or light feeder, how do you feed? So I would like to get more knowledge on that, please.
Sophia, sorry, I missed quite a few because there was an echo. Unless you can type it in, then Erica, or otherwise you must send me a mail. I don't know which is going to be the easiest now.
It's the chicken pellets that we get from Agrimark in Philippi.
Yes.
So we put that.
Oh, the chicken. Yes. What was your question around the chicken pellets, Sophia?
How often do we feed the soil with the chicken pellets, and how much for heavy feeders?
I have to disappoint you, Sophia. I know very little about soil chemistry and fertilizers. No, you're going to have to ask somebody who works in that field. Sorry, I can't answer you. I have no idea.
Okay, Sheila.
Sorry.
Next is Fawaz.
Hi, Sheila. I just want to take this opportunity to thank you for the results. It's obvious because just next to me, I've got like my neighbor got 4 horses. So the manure is like running in here like nothing. So it could be, I saw the result. So I will definitely make a plan on that dry matter. That's my quick answer. But I want to take this opportunity to say thank you very much. Thank you, Erica. Thank you, Cape Town Food Growers. I really appreciate this.
Thank you, Fawaz, for confirming, what I thought. I make statements and it's nice to get confirmation as to why. Otherwise, I think, what have I said? So thank you very much. Appreciate that. Thank you. Thank you.
Hello, madam. How are you?
Thanks. Next is Hafiz.
Hey, so Sheila, I just want to find out with regards to the root knot nematodes and the cis nematodes, and as a matter of fact, for all the nematodes that you found that are parasitic within the root and soil samples that we provided, do these affect nutritional value of crops, not in a sense of lowering nutritional value, but do they have adverse effects on people consuming those crops?
No, they don't have an adverse effect on the crop, but what they do is they compromise the roots, so you don't get uptake of your nutrients.
I
The
see.
plants they'll be weaker and they won't grow as well. That's your
I see.
problem.
So they will be like in this perpetual state of like stress, and they won't know how to defend themselves against like diseases that to like lower the immunity towards other diseases, in a sense.
Absolutely. So what you're going to get yellow plants, and then you think they need nitrogen, so they're going to have yellow leaves, and that's a sure sign that you might have nematodes. There are also other things that affect it. You'll have what we call early wilting. So those roots, as you saw in the pictures, are compromised and they can't take up the water, and you can keep adding water and it's not going to react. It's like those tomato plants, which the tomatoes grew beautifully up to a point, and then they just didn't ripen. They hang there and they don't ripen because the roots can't give the plant what it needs to grow properly. So it's not going to affect the human consumption. It's just you have a poor plant, and then your problems start aggravating. And you just they just add. It's like the last straw breaks the camel's back. That's the problem.
Yes. Okay.
Okay.
This is very, very, very interesting. Very, very enlightening as well.
Glad. We just have to solve your Cis Nematode problem, and I don't know how to do it.
Yes, we will put our heads together and we will do lots of research from here on forth with regards to the Cis Nematodes.
The best thing that you can do, Hafiz, is to tell me which crops you saw on that Cis information that I sent you. Is, we're going to have to look at you planting fewer Brassicas or fewer whatever it is. That's a problem.
I see. Okay. We'll have a meeting on the farm and then see what needs to be done. Yeah.
Okay.
Cool.
Thanks. Nazeer, is your turn.
Thanks, Sheila. I just want to say thank you. Thank you, Erica. I'm so sorry. I had a big problem joining in early on, but I was part. I attended most of the workshop, but I was in a different part of the farm. I just want to say thank you to Erica, and thank you for organizing the soil workshop, hopefully we will continue this tradition. That will be very, very nice to do that, because one of the things that I've learned is that learning is not an event. It's an ongoing process, and we need to be involved in this learning and sharing information over time. And to Sheila, I'm sorry, Sheila, that I put you into all kinds of positions, but it's because because I've a lot of confidence in your work, and I really admire the work that you and your lab is doing, and that's why I direct questions to you, and so I'm really appreciative of your time you take for doing this. One of the things that we realized when we started this fall workshop series in 2017 is that a lot of the knowledge around soil and soil science and soil practice and disease management and all of that is not really coming down into the small scale farming to small scale farmers, and so small scale farmers, they we struggle to get access to the knowledge, and through this workshop and through your Nemlab, we are able to do that. So I really appreciate that. I just got one question for you before I sign off. Sheila, in the overall scheme of things, when we look at all the prevention and management of plant and disease, what role does applying compost and good quality compost do overall in managing pest and disease on the farm? That will be my question. Thank you.
Yeah. Nazeer, you're right. Good compost. That's key. You've got to, it depends on what goes into the compost. So if you're going to put, say, the whole plant with its roots in and all the diseases that go in it, then you've got to compost it properly. Otherwise, you're going to be putting bad stuff back in again. So it's a little bit more complicated. You know, I always say compost is not compost is not compost. It's an old saying. We've been saying that for years in the laboratory. Compost is not easy to make. So you've got to take that into consideration. But again, compost is not the only answer. You know, I went through all those control measures and you've got to look at them more carefully. Rotation of crops is key. It really is. Even on the small scale that you're doing, you can't plant the same thing. You planted your brassicas there, now you're going to plant your potatoes, and now you plant your tomatoes. And every year you do the same, in the same row. You've got to move your rows along or do something like that. So compost is a key component. A lot of other things to look at. That's not going to solve your problems. You know, you've got to have good plant material. I mentioned that. Yeah, there are quite a few things you've got to look at. You can't only rely on compost to do everything. It's not. It definitely is not going to do everything. One, because it isn't the answer for everything, and two, because compost isn't compost isn't compost. And I can say that over and over.
And also diversity is important. So you can use compost, but you have to incorporate the diversity of methods, you know, so you're covering all angles.
Yeah, you've got to cover, you're right, Hafiz, you've got to do your diversity, not only in your actions, but also in your plants. Remember, we have to have different diverse root exudates, because that's going to be the answer ultimately as well.
I think this thing, wow. 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 Yeah.
Can I just come in? Can I just come in quickly, Erica?
Yeah.
Yeah, Erica, I get the sense from some of the attendees that, you know, where do I start? Where do I start? And it seems like so overwhelming that, you know, that all of these things, and then we focus too much on the crisis of, we feel paralyzed when we think about all of these issues that come up when we plant and so on. But I think, and that's why I want to say it's a process. You know, we must plant, we must observe, we must analyze through forums like this and through collaborating with other farmers, share information, and then that process will help us, you know, empower us and give us confidence in improving our production. You know, I want to stress that to everybody, because we think that the information is so much and so technical, and so therefore we're all paralyzed now. We need to become scientists now and get a volunteer job at Nemlabs because we don't understand the stuff better. But really, we need to think about just applying and going on. And every time we learn something new, we apply it, we observe, and so on. And that's why farmers are scientists, you know. And I think we must keep that in mind and not be disempowered by all the technicalities of what we see and what is presented to us. You know, it can easily become that. And that's all I wanted to. I just wanted to note that and observe that and share that with others. Thank you.
I'm going to add to that, Nazeer. It's difficult for me because I give you all these things. It's just a reference. This whole recording is a reference. If you start seeing these problems, you know where to go and find the answer. Most of you don't have problems. None of you did I say you have to treat. So all of you are in a very good position. But as you intensify the growing, so it's going to change. So all I'm saying is, if you start seeing things and be observant, that was the other point I wanted to make. Be observant all the time. and see, and also be aware of mistakes that can happen. None of you have got the problems that you know, there's nothing major that's happened, and you're actually doing quite well. Nazeer is right. Please use this purely as a reference for if you run into trouble. And you've learned to do different things to not get into this problem. Those options I gave you will prevent you landing up being in a problem situation.
Thank you very much, Sheila. There's one last question from Fawaz.
Yeah, thank you, Erika. Just wanted to say thank you to Nazeer Sonday once again for coming so through. I'm sure that is also experience. And thank you for always staying so positive. Yeah, definitely not to be technical. I think it's just to give a little bit more love to them, and then your results would be perfect. So once again, thank you very much, guys.
Pleasure.
Great, and we don't have any other questions at this stage. Thank you very much, Sheila, for your time and for all the preparation and for doing all these tests and the feedback. I'll definitely be in contact with you via email with any other further questions that come through from my side. And thank you for being so open and allowing us to ask all these millions of questions. I'm sure that we can come up with 100,000 more questions and probably another workshop, but I will speak to you offline about that. Thank you everybody for attending, and thank you for your time. I'd just like to invite everybody to please put their videos on.